Hi, thank you for joining us for the Kirby Institute seminar series. I would like to begin by acknowledging the traditional custodian of the land on which we gather today and pay my respects to their elders past, present and emerging. For myself, that is the Gadigal people of the Eora Nation. For Professor Gillings, the Wadamadagal clan of the Darug Nation. I extend that respect to Aboriginal and Torres Strait Islander people joining us today. Some housekeeping to start off with. The format for the seminars will start off with a presentation followed by a Q&A at the end. Q&A is now open. You can interact by typing your questions throughout the talk. When you click on Ask a Question, please remember to write your name. This will help us to reference your questions and make answering easier. You can upload any questions you would like to be answered. You can also change the order of questions by clicking on the tab on the top right of the Q&A chat, which says Most Recent, and changing it to Most Liked. Professor Michael Gillings has a long-standing interest in the generation and maintenance of genetic diversity. He teaches in an introductory biology unit at Macquarie University that has often more than 1,000 students. Michael came highly recommended by my former colleague Gemma Geoghegan, who worked with him in the Department of Biological Sciences at Macquarie University. Michael and I were also involved in a project led by Gemma on virus diversity within foxes. Michael is a distinguished professor of molecular evolution at Macquarie University, and his main interest is genetic diversity and evolution, and it includes a wide range of organisms from viruses and bacteria to invertebrates and vertebrates, but also plants. He's recognized globally for his pioneering work on the evolution and dissemination of antibiotic resistance. His work led to a critical understanding of how environmental and ecology are centrally important in managing the resistance crisis. Specifically, how human activities and pollution influence the dissemination of microorganisms and resistance genes. Today, he will talk about integrons and their role in generating antibiotic resistance. Thank you, Michael.
Thank you very much, Francesca, and thank you for the invitation to talk. So you can see the title of my talk here. I'm going to talk initially about integrons and what they are, and then a little bit about how integrons have participated in the generation and spread of antibiotic resistance. And then I'm going to finish off with a more speculative section on what the properties of mobile elements can tell us about potential means of controlling the spread of antibiotics and mobile DNA elements in general. So let's start with integrons. So integrons are genetic elements. They're commonly found on the chromosomes of bacteria. And the business end of the integron, I hope you can see my pointer here, is this gene here. It's called INTI. It's an integron integrase, and its job is to catalyze recombination of circular gene cassettes that are in the environment into this location here, which is a recombination site. And you can see that if you accumulate gene after gene after gene in this site here, you accumulate an array of these gene cassettes stretching out to the right-hand side, and these are all expressed from a promoter in the integron. So what these integrons are doing is sampling genes from the environment, capturing those genes by recombination, and then expressing them. So how do they work then? Well, integrons are kind of like bacterial sex. So when everything in the environment is stable, integrons don't really do very much. This gene here, the integron integrase, is inactive, and the promoter is expressing these genes here downstream from the recombination site. Let's say we get some kind of environmental perturbation. That can be a heat pulse, it can be a phage attack, a bacteriophage virus attack, it can be a change in pH or oxygen. What that does is it triggers a response called the SOS response. And that then triggers integron recombination. So this gene becomes active and it starts excising some of these gene cassettes and incorporating new cassettes. And what that does in a population of bacteria is generate this enormously diverse sets of cells, each of which has different arrays of gene cassettes in different orders. And that then generates a diverse population of bacterial cells upon which natural selection can act. So it may well be that this particular combination in the second row here is best suited for the new conditions, and that is the cell line that will be selected. All of the others will die. Now, integrons are actually found in lots of different taxa. Here's a kind of a very rough phylogenetic tree of the best known phyla of bacteria. And you can see that if you look at genome sequences, 32% of delta proteobacterial genome sequences and 25% of gamma proteobacterial genome sequences have integrons in them. There are some groups that don't have any integrons, but you can see that integrons are widespread and often at quite high frequencies throughout the entire bacterial kingdom. They're also diverse and ancient. So we can classify integrons according to the amino acid sequences of their integron integrases, the enzyme that does the recombination. And this is just a scratch tree of the relationships between these integron integrases. Originally, when integrons were first discovered, they were assigned to classes 1, 2, 3, 4, 5, etc. But after class 5, so many new integrons were being discovered in the chromosomes of bacteria that there's now too many of them for this class's naming system to continue. So these systems, the integron systems, they've been operating in bacteria for possibly billions of years. So now that we know what integrons are doing and how they operate, I want to just step back a bit and think about the most recent part of evolution, which is humans and their activities and how we've affected the biosphere. So humans are now the world's greatest evolutionary force, and we have dramatic impacts. Our activities have impacts on all Earth processes, all biogeochemistry, and on all other organisms on the planet. And the kinds of things we do are listed on the left-hand side here. We compete with other organisms by using resources in space. We kill things, so we've precipitated the 6th mass extinction, for instance. But the two that are of most relevance to this talk are pollution and selection. So we pollute the world with bioactive compounds that affect the activities of other organisms. And we also impose selection on all sorts of organisms, and that accelerates evolutionary change. So we are driving evolutionary change in all organisms on the planet. Now, sometimes that drive is direct in circumstances like domestication, or when we hunt things, or when we harvest fish from the ocean, or when we use biocides to control the growth of other organisms. We also drive evolutionary change indirectly through our other activities, like rising carbon dioxide that leads to ocean acidification, climate change, shifting climatic zones, and through our release of pollutants. And all of this leads to evolutionary change in organisms, of which antibiotic resistance in bacteria is probably the clearest example. So resistance to antibiotics in bacteria appears really quickly. Generally, if you introduce an antibiotic to clinical practice, it's less than a decade later that resistance to that antibiotic becomes a clinical problem, and it spreads between different organisms. So you can see that here, every time you introduce an antibiotic, let's say ampicillin here, resistance appears fairly shortly afterwards. The most recent antibiotic classes, daptomycin, for instance, the resistance appears really, really quickly, like matters of years after the introduction. So how does this resistance happen? Where does it come from? Well, there are two ways of acquiring resistance. The first is by mutation. So antibiotics work on bacterial reproduction by targeting particular elements of bacterial biochemistry. They have target sites that they bind to or inhibit, and you can generate antibiotic resistance by mutation. So mutation to the target site changes the way the antibiotic works, and it no longer can interfere with bacterial biochemistry. The problem with that is that mutation only occurs in the cell that it originally occurred in and its descendants. There's a faster way of acquiring resistance, and one which can spread much more rapidly, and that's lateral gene transfer. Bacteria can acquire genes from the general environment, test them out, see if they confer resistance, and if they happen to confer resistance to an antibiotic, that fixes that lateral transfer event into the cell, and that cell goes on to found the new populations of resistant organisms in an infection or in a hospital or in an environment. So lateral transfer is important. It's not that mutation is not important, but lateral gene transfer is potentially more important. And the reason for that is because it confers immediate changes to phenotype, and it can spread those phenotypes between different species. So let's go back to integrons. Remember, integrons can acquire gene cassettes from the environment. And some of the genes that it can acquire are going to be genes that can confer antibiotic resistance. Now, integrons then are sampling these diverse genes from the environment. They're exquisitely preadapted to acquire resistance genes. So they're sitting there waiting for humans to come along. I mean, that's teleological, but you get the idea. One of the things about antibiotics is that they cause environmental perturbation. So antibiotics trigger the SOS response. The SOS response then turns the integron on. It starts to acquire all sorts of gene cassettes from the environment, and then the antibiotic is still there to provide the selection to fix resistance genes in particular bacterial lineages. So you've got a population of bacteria with an integron, add antibiotic to it, switches on the SOS response, the integron starts sampling all sorts of different genes, you end up with a diverse population of cells. One of those cells might have acquired a gene cassette via integron activity that confers resistance to antibiotics. So integrons were always going to be a problem as soon as we started using antibiotics to control bacterial growth. Now, of the many integrons that you can see in this phylogeny here, which of these integrons, of these many integrons, acquired genes for antibiotic resistance? Well, it turns out, this is a heat map along the top showing the number of antibiotic resistance genes found in each one of these integron families. Turns out it's mainly class 1 and class 3 integrons with a few class 2 integrons. And here, this recapitulates the history of antibiotic resistance. The reason this is called class 1, class 2, and class 3 integrons is because they were the first 3 integrons to be discovered, and they were discovered because they were causing problems generating antibiotic resistance in clinical pathogens. So why is it that class 1 and class 3 integrons seem to have most of these antibiotic resistance genes? Well, firstly, their integron integrases, the things that do the recombination and gene cassette acquisition, are possibly more flexible. and they recognize more recombination sites in gene cassettes from environmental compartments. So for instance, here are some of the recombination sites folded up into a secondary structure, and you can see there's lots of diversity in shape. Turns out that class 1 and class 3 integrons are more flexible in recognizing each one of these kinds of recombination site. So that's the first reason. The second reason is because class 1 and class 3 integrons became associated with mobile DNA elements. That allows rapid spread of these particular integrons between different species and different cells. Most integrons are on chromosomes. So if you acquire a resistance gene, it gets incorporated into your chromosome, but it can't go anywhere else. It can't get into other species. And then I think the last reason is chance. They're the first to acquire resistance genes. It's just a stochastic thing. And if you're first off the rank with, you know, a new resistance gene, and you inhabit all of the places where that antibiotic is used, other resistance genes don't really have a chance to overtake you because you're already at high numbers. Okay. Now, to spread rapidly, the integrons that were capturing these resistance genes needed to be mobile. They needed to be able to move between bacterial cells. And that's what this slide is about. And I have to say that this slide represents and summarizes about a decade of work, which seems ridiculous to me now, but, you know, science is slow and hard sometimes. So this is the reconstruction of what we're pretty sure happened in a critical time during the development of antimicrobial compounds. You've got diverse class 1 integrons on chromosomes. Some of these integrons, or one of these integrons, got captured by a transposon called TN402. Transposons are genes that can move around between locations in bacterial chromosomes. That generates this kind of structure here. That structure itself was then inserted into a mercury resistance transposon, that's this thing here, to generate a compound transposon that itself was sitting inside a plasmid. And the integron is active and it's able to capture lots of integron gene cassettes from the environment. Now, we think that this event, this complex event involving, what is it, 1, 2, 3, 4, 5, 6 maybe different mobile elements, happened once, and happened once in a single cell. The reason we think that is because clinical integrons, this sequence here of the integron integrase is 100% identical or nearly so in all clinical class 1 integrons. This is what the first mobile class 1 integron looked like. We can still find this element in environmental samples. So we originally found it on the leaf surface of baby spinach, and from that leaf surface we recovered a strain of Enterobacter cloacae, which is an interesting organism because it has strains that are adapted to living on plant surfaces and other strains that are adapted to living inside the gut of warm-blooded animals. So this element here consists of a mercury resistance transposon, inside of which is another transposon, inside of which is a class 1 integron, and that class 1 integron has a number of interesting genes, an NADPH-dependent flavin mononucleotide reductase, a quark family gene, both of which are cassettes, and down the end is a mercury resistance operon. By the way, you might notice that we sequenced all these plasmids, and because we found it on baby spinach leaves, we call it POPI, P-O-P-I, with using the standard nomenclature for plasmids, but no one noticed until it's through the review and et cetera, et cetera. So POPI is the plasmid inside the Enterobacter cloacae that is on baby spinach leaves. So, oh, wait a minute, what have I done here? I've made this big somehow. How have I made it big? Yeah, let's just see here. Not sure how I'm going to go to the next page here. Oh, I'm having technical problems. What is going on here? Just needs to be shrunk.
I'm not sure how to do that, but you could try and press escape just to get out of that.
Okay, there we go. Yeah, awesome. Thank you very much.
No
Okay,
worries.
so remember how I said that these genes here are really interesting. They're resistance determinants and they're of particular interest. So why is that? Are they of particular interest? Here we go. Turns out that these three genes confer resistance to early antimicrobial compounds. The first of these repairs the damage done to proteins by arsenic, which was an early antimicrobial compound used to control mainly sexually transmitted diseases. The next of these, Quoc-E, that is a membrane efflux pump. Now, they're proteins that sit in bacterial membranes and pump things out of the bacterial cell. It turns out that this particular membrane pump can pump out benzalkonium chloride, which is a disinfectant still used in things like Dettol today. And finally, the mercury resistance operon confers resistance to mercury, which again was an early antimicrobial compound used again to control mainly sexually transmitted infections. So sitting here, we've got an element, a mobile DNA element with an integron at its core that confers resistance to all three of the very early antimicrobial compounds that were in use in the beginning of the 1900s. So we think that this element probably fixed and became a thing somewhere in the 1930s to 1940s. We'll never be able to prove that, but that's what we think. Now, of course, this integron is still active. It can still do things. So it can acquire diverse resistance cassettes. Every time you introduce a new antibiotic, the integron can fish around in the environmental DNA pool for gene cassettes that confer resistance to the newly introduced antibiotic. The transposon here, TN402, is a really interesting transposon. It's called a res hunter. And what that means is it hunts resolution sites of plasmids, and so it inserts itself into diverse plasmids. So the integron is working and grabbing diverse resistance gene cassettes from the environment. The transposon and complex mosaic element that you see here is inserting itself into lots of different plasmids and transposons, and those different transposons and plasmids are moving between diverse bacterial species. So that led to rapid expansion of this mobile class 1 integron. We think that there was a single copy of this thing in a single cell in the early 1900s, and now integrons derived from that original event have acquired a total of 130 different resistance genes to antibiotics. They've spread to lots and lots of different broad host range plasmids, which are DNA molecules that can move between bacterial cells of the same or different species. It's spread to at least 70 species of bacterial pathogen and an unknown number of environmental organisms. It's spread to every continent, including Antarctica, and it's now universal in the microbiota of humans and our domestic animals, agricultural animals and companion animals. So that we all now have resistance genes and class 1 integrons in our microbiota. Every person listening to this talk has class 1 integrons in their gut bacteria. And the diversity of resistance genes that's associated with our gut bacteria increases as we age. This is children, school children, high school students, and adults. As we're exposed to more and more antibiotics, more and more other people, more and more environments, as we eat more and more diverse foods, we accumulate antibiotic resistance genes and integrons in our gut. That means, because, you know, if you've got things in your gut and on your skin and in your mouth and your nose, we shed those into the environment. So we shed integrons into our own homes. And we've been showing this by testing dust samples from vacuum cleaners. So if you get the HEPA filter from a vacuum cleaner and shake it out and extract DNA, you just do PCR for integron integrase. This is the result from my lounge room and spare bedroom. So, you know, there's integrons in where I'm sitting right now in my lounge room. And 95% of vacuum cleaner dust samples from all over Australia are positive for this gene. So in 2017, I decided to do a little calculation about exactly how many copies of class 1 integrons were being shed every day by humans and their domestic animals. And it's a pretty simple calculation. We know from qPCR results how many copies, or the range of copy number in feces of pigs, chickens, cows, humans, goats, horses, etc., etc. We know how many grams of feces each of these individual animals produces per day, and we know the size of the population of each of these animals. You just multiply the three together and you get huge numbers out the other end. So this is the total copies of this particular gene released every day. And you can see from pigs, it's somewhere between 10 to the 19 to 10 to the 23 copies per day. Bear in mind that this particular formation of this gene, this particular arrangement of it that is being released in these copy numbers, was a single copy sometime in the 1930s, probably. So this is per day, of course, and these copies have a half-life in environmental circumstances. So we're adding to these numbers every day because the decay rate is nowhere near as fast as the rate at which new copies are being added to the environment. So I did a meta-analysis of all of the qPCR studies that have been done using this gene as a target, and stratified it according to the kind of environment: soil, manure, wastewater treatment plant sludge, effluent, rivers, aquatic sediment. And you get a range of numbers, but the important one here is this red line represents one copy of this gene INTI1 per bacterial cell in the environment. If we break this down and actually look at the raw data, they look like this. So you can see the lowest abundance, as you might expect, is in Antarctic soil. You can see that the highest abundances are in chicken manure, but also downstream from pharmaceutical plants. So pharmaceutical plants often release their waste liquor from fermentations and from production facilities directly into the waste stream. So in some cases, in places where you know, the pharmaceutical industry is not as controlled as it is in Australia, they're releasing huge amounts of antibiotics directly into the environment. And it's no wonder that there's enormous quantities of class 1 integrons downstream from this particular pharmaceutical plant. But the other thing you'll notice here is that wastewater treatment doesn't really do very much to remove this gene, because wastewater treatment is about removing bacterial cells and killing them, not about removing DNA. So if you look, here's wastewater, the proportion of this gene in wastewater. This is in sludge. This is the effluent coming out of wastewater treatment plants. This is rivers and lakes and freshwater sediment, which are the final repositories of wastewater effluent from treatment plants. We think that probably the pre-industrial range of concentrations of this particular gene is somewhere down here. What is that? 1, 2, 3 orders of magnitude below what it is at the moment. So what are the potential consequences of this? Because we've got vastly increased abundance of this class 1 integron, and it's mobile, and it's in the environment. It's being released into the environment all the time. So I thought that this would increase the likelihood that it would transfer into environmental bacteria, because you're releasing the integron with its antibiotic resistance genes and metal resistance genes, and you're releasing metals and antibiotics in the same waste stream. Environmental bacteria that are able to acquire this integron are going to have a selective advantage. So lateral transfer into environmental bacteria. That in turn means that these things are going to be acquired by wild animals. And of course, the integron is still active. So it's still acquiring novel gene cassettes with selective advantages. And it's likely to participate in the generation of more and more complex pieces of DNA that I have called xenogenetic in parallel to xenobiotic. So the xenogenetic DNAs are DNAs that have been assembled by the actions of human selective agents. So these would have never appeared on the planet but for the actions of humans. So now I'm going to tell you a little story that ticks all of those boxes, and it's this one. It's Integrons and the Eastern king prawn, which I kind of think sounds like a Harry Potter movie or something. I'm not quite sure. Anyway, so up the top here, we've got a pretty standard class 1 integron from a human going into the environment. Of course, the integrase is still active, and this integron has picked up a really interesting gene cassette, MSRA and MSRB. These are methionine sulfoxide reductase genes. Now, what they do is they repair the damage to proteins caused by oxidative bursts from vertebrate immune systems, and from invertebrate immune systems as well, I should add. So, you know, when you're infected with a bacterium, you do this oxidative burst, you damage the methionine, that damages the proteins in the bacteria and kills the bacteria. This gene cassette actually repairs that damage. So a pollutant integron here has acquired a new gene cassette. It's also acquired a new mode of mobility. This is a miniature inverted repeat transposable element. And that whole thing has then inserted itself into some plasmids that we recover from the gut of king prawns, where the plasmids reside in 3 different Acinetobacter species. And as you all know, Acinetobacter is an emerging pathogen. So the previous slide, I was talking about these things. There's been lateral transfer into environmental bacteria, that is Acinetobacter sitting in the gut of king prawns. The king prawn, of course, has acquired those Acinetobacter species that contain those plasmids. There's acquisition of novel gene cassettes that have selective advantages because they protect bacteria against immune responses of animals. And the integrons have participated in the generation of more and more complex xenogenetic DNAs. So all of the predictions that I made in the previous slide have actually come true. One prediction that I made that hasn't come true is I'm expecting to see these species of Acinetobacter, which don't have species names yet, as emerging nosocomial pathogens. I'm glad to say that that hasn't happened yet, although, again, I also have to say I'm a little bit disappointed that it hasn't happened. I mean, just from a perspective, predictive point of view. So these integrons, we've now characterized the plasmids that they've got into, and they're participating in diversifying these megaplasmids that are in Acinetobacter. These megaplasmids are chock full of antibiotic resistance genes, as well as the integrons they've now captured. other thing is that these integrons that are being released in human waste streams and in agricultural waste streams have been acquired by wild mammals. Here are 3 examples. This work has been done by Michelle Power, one of my colleagues, brush-tailed rock wallabies, flying foxes, and Australian sea lions, some of which are endangered. And interestingly, they've acquired gene cassettes, which we have not seen in clinical practice yet, but are antibiotic resistance genes. Okay, so, you know, integrons have had this central role in antibiotic resistance, in spreading antibiotic resistance, not only amongst bacteria, but amongst hosts, animal hosts. They're spread into environmental bacteria, they've spread to every continent, and there's huge numbers of these in the environment. And that vastly increases their probability of finding new gene cassettes. So one of the questions I was asking was, well, what kind of gene cassettes can they access? And could those gene cassettes be of concern to humans? You know, what else could they code for besides antibiotic resistance? So you can survey gene cassettes in environmental samples because they have a comparatively simple structure. They have a recombination site and an open reading frame. That's pretty much all they are. So there's the gene that encodes the protein, and there's the recombination site. The recombination site is semi-conserved, and you can design PCR primers to the recombination site, use that PCR on environmental DNA extracted from soil or biofilm or sediment, and that will amplify gene cassettes. So if you do that in environmental samples, it generates multiple products, each one of which corresponds to a different gene cassette encoding a different protein. So here's a really early experiment we did. This is Urandary, it's a silver mine, and it's an acid mine drainage site. We just took a transect across here and took soil samples. You can see the sulfur in this soil. Every 1 metre, you extract DNA from all of those soil samples across the transect, amplified gene cassettes, and this is the pattern that you get. Each one of the bands on this gel represents a different gene cassette encoding a different protein. And you can see there's enormous spatial diversity here. I mean, there are some adjacent samples that have similar patterns, but in general, there's, you know, the impression that you get from this gel is that there's lots and lots of diversity here. So we wanted to ask, how much diversity? How many gene cassettes are there? So what we did, we collected samples from diverse locations, from Antarctic soils, from the Macquarie campus, and from the Sturt National Park in far northwestern New South Wales. It's a desert. Amplified gene cassettes from each of 48 soil samples and Illumina sequenced them. We recovered somewhere around 45,000 different gene cassettes. They encoded 27,000 unique proteins. And we discovered that each 0.3 grams of soil, which is about the size of an aspirin tablet, has somewhere between 4,000 and 18,000 different gene cassettes. So these gene cassettes are a repository of enormous functional and genetic diversity. Then we ask questions about the biogeography of gene cassettes, and Gemma Geoghegan is actually responsible for generating this beautiful graphic here. So each column is a different location. Each horizontal line is a different gene cassette, and the colours represent the relative cassette abundance of a particular cassette per million reads of Illumina sequencing. First thing that you can see is that the most common gene cassette in every single sample, the most common set of gene cassettes, is unique to that particular sample. So this shows that gene cassettes exhibit really, really strong spatial structuring down to scales of 1 metre. That means every metre that you go, and we already knew this from the previous gel that I showed you, every metre that you go, you get a completely different set of gene cassettes, which is really amazing and bizarre. Lots of gene cassette types, in other words, the protein encoding gene cassettes, only occur in a single sample. there's a very small subset of gene cassettes that occur in every single sample, regardless of whether it's a desert, Macquarie campus, or Antarctica. So there are a set of gene cassettes, there's about 600 of them, that occur in every single sample that we look at. What we think is going on is that gene cassettes are being generated locally. I mean, the formation of gene cassettes is still an open question that we're working on, but we think that this strong spatial structuring and strong uniqueness of gene cassette profiles at a single location represents newly formed gene cassettes that are blinking in and out of existence. They're being formed and lost at particular locations, every particular location you look at. There's a little bit of local dispersal and selection, and then some gene cassettes go on to have global dispersal because they confer some sort of advantage to the cells that carry them that applies regardless of where it is, whether it's Antarctica or Macquarie or the desert. So we need to think about these biogeographical patterns when we're thinking about how bacteria respond to control strategies. And so what we thought about doing was predicting the future. So we know that there's lots of class 1 integrons in environmental samples. We know that the huge numbers vastly increase the probability that they're going to require the gene cassettes that are globally dispersed, that are really common and are everywhere. So what do these gene cassettes encode? Well, there's about 600 different types. They're found in every sample tested that we've tested so far. The functions of the cassettes are largely unknown. They're hypothetical proteins or conserved hypothetical proteins. There's a few that are identifiable. You can see the proportion here. So we asked, well, of these identifiable protein families, of these universally distributed gene cassettes, are there any that we should be concerned about? And the answer, of course, is yes, there are. So phenotypes of concern are identifiable. The first, as we would have expected, are antibiotic resistance determinants. Beta-lactamases, N-acetyltransferases, dihydrofolate reductases that confer resistance to trimethoprim, bleomycin resistance. But there were some other things that are equally of concern. So there are type 2 secretion systems, virulence factors, and glycosyltransferases. Each one of these, as a cassette, could potentially increase the virulence of the bacterium that captures the cassette. Then finally, one of the solutions to the antibiotic resistance crisis is to use bacteriophage to control bacterial growth. Well, unfortunately, there are also cassettes that encode phage resistance. So there are cassettes that encode CRISPR-Cas3, and there are cassettes that encode methylases that can methylate DNA and prevent it from being cut up with restriction enzymes. So if we start to use phage to control bacterial infections, I guarantee that integrons are going to capture phage resistance genes and protect the bacterial pathogens from phage therapy. Okay, so there are other concerning trends though, because, you know, the integrons are doing one thing, they confer a phenotype, and most of our clinical approach has been about the bacterial species. You know, you identify a species and you use an antibiotic to kill it. Well, what that does, it promotes acquisition of resistance genes. You can concentrate on the phenotype. You can say, well, what kind of resistances are here? And you can choose antibiotics that your target organisms are not resistant to. Well, that promotes acquisition of multiple resistance genes eventually. What we could do instead is focus on the mobile elements themselves, because it's mobile DNA elements that rapidly spread these phenotypes of relevance to human and animal health. Can we think about ways of controlling the mobile elements themselves and use that to damp down the rapidity with which resistance of all kinds accumulates and spreads within bacterial populations. New ways of thinking about mobile DNA could help. So first, we need to stop thinking about these as DNA molecules, and we need to start thinking about them as ecologically and evolutionary independent units of life. These things replicate. They replicate in the best places for their replication. So for instance, a lot of mobile elements have what's called conjugation systems. These conjugation systems allow the mobile element to move between a donor cell to a host. These conjugation systems aren't actually beneficial for the hosts themselves. And plasmids with conjugation systems are like rats fleeing a sinking ship. If they detect that their host cell is about to die, they turn on their conjugation systems and move to another cell. We also could think about these mobile DNA elements as environmental pollutants, and we can also think about them as invasive species. So, DNA is a pollutant, and it should join other pollutants as a cause of concern. So I've defined pollution fairly broadly as the dissemination of materials that could have harmful effects. We've got conventional pollutants, crude oil, nitrates, carbon dioxide. We've got xenobiotic pollutants, which are things that humans make, radionucleotides, pesticides, plastics, synthetics. We've got invasive species here as a pollutant as well. And then we've got xenogenetic elements, these DNA elements that have formed under human selection pressure. And you can think about these in a box like this, kind of like a Punnett square or something. Pre-existing compounds, conventional pollutants and invasive species, novel pollutants, which are xenobiotics and xenogenetics. And if you look at the horizontal axes here, some pollutants have finite quantities. So you pollute, they have a half-life, gradually the pollution disappears. But invasive species and xenogenetic elements are able to replicate. So these are pollutants that replicate. And they interact. So each one of these other pollutants increases the likelihood for spread and fitness and abundance of xenogenetic elements. So maybe we could think about controlling mobile DNA in the same way that we control invasive species, because lots of ecological and evolutionary work has been done on invasive species and how to control them. And the 4 strategies are these. Quarantine, that is stopping things from coming in. Well, xenogenetic elements arise spontaneously and they spread really easily. We could repair environmental damage, and that would work. We could remove selective agents from environments. That would take away the advantage that these mobile elements have, and gradually they would decline in abundance and in distribution. We could detect these things early and try and eradicate them, and certainly we can do that with better technology and with environmental DNA monitoring of things like sewage, for instance. Or we could use biocontrol elements. And I'm still thinking about this because I want to try and think about what a DNA biocontrol agent would look like. It's kind of hard to think about. So in summary, humans, we've attempted to control bacterial growth because, we don't want bacteria to infect us and kill us. But those attempts to control bacterial growth have had adverse consequences. We've selected for resistance genes, and potentially we're now selecting for virulence genes and genes that increase transmission and survivability and phage resistance and all of those kinds of things. We've certainly selected for increased mobility of these resistance genes. And along the way, we've driven the assembly of these mosaic DNA elements that have multiple resistance genes and multiple bits of DNA from all sorts of different sources, different phyla, different species, different places. We've seen rapid increases in the distribution of all of these elements, mobile DNA elements and resistance genes, and in the abundance of these mosaic DNAs we have helped create. And that's led to widespread pollution with mobile DNAs and their host cells. And potentially that's led to an increase in the rates of bacterial evolution, simply because we're ramping up the selection pressure that we apply to bacteria. So these are all problems that I'm grappling with at the moment. I'd like to think about some solutions, but you can see that the solutions that I'm suggesting involve lots of different fields, lots of different areas, lots of different research arms, only some of which, in fact, a minority of which I have expertise in. Okay, so thanks for your attention. I hope you've enjoyed the talk. I'm more than happy to answer questions if I can. My lab is called the Emma Laboratory. Here are some of the people who've helped in generating these data. Thank you very much for listening, and I'll hand back to the moderator to do the Q&A session. Thank you very much.
Thank you, Michael. Great presentation. And we have a couple of questions. First, a comment from the audience. Fantastic, fascinating presentation. I think I can agree. I have learned so much about.
Thank you. Thank you very much.
We start off, maybe we can combine these two questions, and how are gene cassettes generated? And is there any particular pathway that these integron mobile elements seem to transfer between species? For example, conjugation, secretion systems, environmental DNA uptake.
Okay, so the origin of gene cassettes is still a mystery. We suspect, because gene cassettes just consist of a recombination site and an open reading frame, that they're actually a reverse transcription product of some sort. But we don't know. No one has ever observed a gene cassette being formed. No one has ever really put forward a testable hypothesis. I mean, people have been working on this for a long time, and we currently have an ARC discovery that, you know, we've said we're going to try and find out how gene cassettes are formed. That is a pretty big ask, and we'll see how we go. You know, watch this space. Maybe in 2.5, 3 years, I'll be able to tell you how it happens, but who knows? Are there any particular pathways? Well, if what I'm thinking is true, if these gene cassettes are generated by reverse transcription, then essentially any gene anywhere in the biosphere could become a gene cassette. And when we look at gene cassettes and try and figure out what they are, maybe 80 to 85% of them are orphans. They are open reading frames that have never been described before, that don't have any homologs in protein or DNA databases. So they are the first record of that particular sequence. So gene cassettes in general are this repository of enormous and undiscovered genetic diversity. It's kind of like the dark matter of the microbial DNA world. So, you know, we can identify resistance genes because a lot of work has been done on them, and a lot of, you know, phenotypes have been examined, and also the, you know, the structure and function of these products has been elucidated. But the vast majority of gene cassettes are a big black hole. We don't know what they do.
Thank you. We have two questions from Michael. One would be, so integrons seem highly advantageous. Why are they not universal? Do bacteria without them live in highly specialized niches?
That's a really good question. So integrons are actually quite expensive to run. They're better than transposons, because the integron itself is a site where the new genes are inserted. So it doesn't interrupt necessarily pre-existing genetic or biochemical networks. And, you know, you just express the gene and see whether it does something good. If it does, you retain it, and the cell that carries that has an advantage. we know that particularly for places where environments are fairly stable, integrons tend to degrade fairly quickly. So for instance, Xanthomonas, which is a phylosphere leaf surface specialist, has integrons. But when you're a Xanthomonas and you specialize on a particular species of plant, like tomato or citrus or geranium or something like that, the integron activity tends to be lost. So you have your gene cassette, but the integron integrase tends to accumulate deletions and stop codons and eventually gets deleted altogether. So there is a problem with maintaining integrons and integron activity if you don't have to deal with a changing environment that's subject to perturbation.
Thank you. And follow-up also from Michael. And can an acquired gene be lost if it ceases to be advantageous? And I might have a follow-up question to this. If you talk about the mercury resistance and in relation to using mercury as a therapy for sexually transmitted disease, which as far as I know, it's not used anymore.
No, it's not.
Then bacteria would lose that because it does not give them any advantage anymore. Yeah.
Okay, so to deal with that question first, that is absolutely true. So the right-hand end of that element that I showed you before has often been lost. So you lose the mercury resistance transposon, you lose a lot of the original TN402 transposon as well. So now the integron is kind of stuck in the plasmid that it finds itself in. The first question, which was about losing gene cassettes if they're no longer of value, yes, because what happens is as you push the gene cassette further and further away from the integron, let's say you incorporate 6 cassettes, by the time you get to you get pushed to cassette number 7, the promoter is no longer expressing that gene. It's too far away from the promoter to be reliably expressed. So most integrons that deal with antibiotic resistance, the maximum number of gene cassettes that's ever been found is 8. And usually it's more like 2 or 3. But in some particular species or genera, like Vibrio, for instance, you can have up to 200, 250 gene cassettes all in a row. Now, we suspect that there are promoters embedded within that array that keep all of those gene cassettes active and expressing. But every time you undergo an environmental perturbation, some of those cassettes are lost because you can get recombination between cassette recombination sites that excises a whole chunk of gene cassettes. So you can lose gene cassettes as well as gain them, is the short answer.
Thank you. I have many more questions, but because of time and me being very Swiss, I just have one last question, and it's, do you take antibiotics yourself?
I try to avoid taking antibiotics wherever possible. Oh, the other thing is that, and probably too much detail here, but one of the main problems with antibiotics is that 70 to 80% of the antibiotic that you take is excreted through the kidneys unchanged in urine. So, and you should know this because when they first introduced penicillin, it was very, very expensive, and they used to collect the urine of the people they treated with penicillin and repurify the penicillin from the urine and give it to the next patient. So all of that goes into sewage treatment plants, and antibiotics are small molecules that can actually survive sewage treatment. So there are 2 solutions to this. One is that we should have toilets that separate feces from urine, and the urine should go to a separate treatment facility. But the other thing that you can do, which is easier for me than it is for you, Francesca, is when you're on antibiotics to wee in the garden, right, onto soil, because it's not going to do any harm there and it'll get broken down. Also, it's probably good for plants, not the antibiotic bit, but the nitrogen bit, yeah? So if I do have to take antibiotics, and sometimes I do because I prefer to remain alive, I try and wee in the garden.
Okay, good.
Yeah.
Thank you so much, Michael. This is a really
No
great
worries.
presentation.
If anyone does have any questions, burning questions that they need answered, just email me and I'll get back to you as quickly as possible.
Thank you. Thank you.
Okay. Thank you very much.